Okay, so I asked you, yeah, anybody recognize anything up here? Just looks like a thing, you know? I would say shark. People like shark. There's our early cartilaginous fishes. Believe it or not, they're not close relatives of today's sharks. I know that might seem ridiculous because of what they look like. And then there's the big guy here in the middle. This is one of these paleo celebrities that even people who have like Dunkleosteus. There was a paleontologist back in the day. His name was Dunkel. So this is Dunkel's Dunkleosteus. That's where that name comes from. And this is one of these animals that's like occasionally in a toy set or like, in a movie kind of thing. So it's fun to think about. We'll talk about it. Let's get into gnathostomes today. So the jawed vertebrates. So just to refresh, when it comes to lecture, we've now well passed to within vertebrata. You guys have spent a little bit of time now seeing a lot of the early part of the Paleozoic vertebrates. Almost all of those vertebrates are jawless fishes. We might call them agnathans, meaning they don't agnath, they don't have a jaw. Gnath is a jaw. Gnathostomata over here, that's jawed mouths. But that's kind of like a descriptor of them. It's not a clade. Many of them have varied relationships. Some of them are related to today's jawless fishes. Most of them are on the stem of our clade, the jawed vertebrates of today. And so animals like osteostracans, you guys saw that little model in lab. Was it cute? Did anybody break it more than it's already broken? Okay, good. But I like that little thing because when you actually see it and hold a model, I mean, it's just a model, but still you can get a sense of it. A little osteostracan like that is a jawless vertebrate, but it's our sister in terms of jawed vertebrates sister. It's not more closely related to any of these other jawless, I'm sorry, forms. And what I hope you can see is that so many of the characters that make up the jawed vertebrates are kind of spread out. And again, that's the real power of the fossil record is like, if evolution is operating how we might expect it to be operating, we should be able to find fossils to show us like the building blocks of getting to an inherited anatomy that's very complicated, but spread out over time and space. And so here is a modern vertebrate phylogeny. You'll see I have 3 synapomorphies there for the vertebrata node and 8 synapomorphies there for the gnathostome node. 1, 2, 3, 4, 5, 6, 7, 8. Those are the same 3 and the same 8 as are on this slide. But they're spread out if you think about fossils. If you're somebody who's only looking at today's biodiversity, certainly the jawed vertebrates do seem quite different than the jawless vertebrates that are all alive right now. You guys now have the benefit of understanding that there's fossil forms in here that help us get an understanding of like pectoral fins comes way after a mineralized, dermal skeleton. That's cool. I think that's cool. And jaws is one of these last things actually to evolve after a dorsal fin, after a pectoral fin, after a head or a circle tail. I think that pattern of relationships is really important. You guys are again represented by the magpie on here. Just for fun and to kick us off, because I haven't seen you in a minute, talk to your neighbors. Do you know all these fish? I know that you don't, please. I'm kidding almost. But like, who do you know? And talk about their relationships to another. Because remember, you can follow the relationships back. So who's our closest relative? And then how are we related to other ones that you might actually recognize? Just try to articulate that a little bit, just to warm up.
Large bass, proud Don, see the temple. Well, they are so actually in the same. It's about it. This route, no idea what that to do with it is there. So the lines that are talking about this certainly down. Don't think we did they said that. Yeah, I think when he's so very common. It's not very awesome. This one was clear. I don't know that answer mom. Actually, I think the one is sure just to you put like I don't I think those are the part of there is I've never seen them on this. What's the sound?
So just to be really clear, I think it would be completely normal for you to be like, I don't know any of these animals. Maybe the magpie, maybe the hammerhead shark. But otherwise, right? Fish are fish. People usually think about fish as like this, like, fish. It's just, it swims. I get it. There's so much fish. Most vertebrates species are fish today, even. And so that's like really, it's actually getting close to 50-50. But Still, a lot of vertebrates and fishes, a lot of fish biodiversity. What are some things you guys are talking about as you look at the phylogeny here of these fishes? Yeah, Trevor.
So with the ones at the bottom, like that, at the bottom, right, I guess.
Yeah,
So it's
yeah, yeah.
the cartilaginous species.
Yeah, so you might know a little bit about some of these animals already, and it's definitely fine if you don't. That's why we're in this class. But yeah, these three here are all in this group that are the cartilaginous fishes. They don't have like true bone in their skeletons. They do have skulls and vertebrae, but they do not have bone to make up those skulls and vertebrae. So that's cool. Just like these animals don't really have a lot of bone in their bodies either. Interesting. Who's our closest relative? Kind of these weirdo fishes. These are some of these lobe-finned fish. We've talked about them a little bit in our lecture, or sorry, in our lab. You guys saw the coelacanth and the trout over there when I put it with a cow. Could have done the same thing with a magpie back in lab, but I used a cow because it's funnier. And that's the example they did in the 1950s when they were talking about this the first times. Over here, though, this is where most vertebrate biodiversity really is today in terms of in one big clade. Over here in this ray-finned fish group, I call those like the fishy fishes, the one that you mean when you say fish. Bony ray-finned fishes like trout and seahorses and coral reef animals. Those are almost all in this group, Actinopterygii. And so we're going to talk about all these here in a second, but I wanted you to kind of get in there. You can see now why people make these like memes and t-shirts and stuff about how like you have a fish. There's books and PBS specials called Your Inner Fish. So much of your anatomy is inherited through vertebrate evolution, and so much of the early branches of vertebrate evolution is fish stuff. It's a lot of fish stuff we have to talk about in this class. So here's some taxonomy for you guys. You already have seen this. I gave you that slide a couple weeks ago now where it was only 5 animals. 1, 2, 3, I think it was this one, 4, and 5. So one side was 3 gnathostomes, the other side was 2 cyclostomes. And so that's still true here. So vertebrata is a monophyletic clade. It has 2 sister lineages. Those sisters are the cyclostomes and the gnathostomes. Gnathostomes are those jawed vertebrates. Once we get to Gnathostomata, there's another one of these sister group splits, and hopefully you'll learn these and get pretty comfortable with them. Osteichthyans and chondrichthyans. And so ichthyes is fish. You maybe have heard of like ichthyology or something. Oste is bone. Chondra is cartilage. So it's pretty easy, I think. Once you have a jaw, you're either a bony fish or a cartilaginous fish. And again, these are names we get from the modern living biodiversity. It's fun how fossils map onto this. Once you're up there with an osteichthyans, we'll break up chondrichthyans later. Once you're up there into osteichthyans, there's another way we can break them up. That's super valid. People could observe this pre-evolutionary tree days, but we of course have DNA and all kinds of other reasons to now know this is how they're related. You can break up osteichthyans into that word, which I love, you might not love, but I hope eventually you will love, actinopterygians, and the other side is sarcopterygians. The pterygi is referring to like a wing structure, the fin. And then actin is talking about the ray structure of those fins, and sarcop is like talking about like muscles. It's this prefix, I guess that you'd say comes from like a muscly thing. These have fleshy fins, and these have, anybody seen a goldfish in a tank or a betta fish, right? They have these fins, but it's like there's these rays that have like a curtain between them, and that's how the structure works. So vertebrates into cyclostomes and gnathostomes, gnathostomes into osteichthyans and chondrichthyans, osteichthyans into actinopterygii and sarcopterygii. You can practice that as much as you want at home. You have these slides. If you want to help yourself get used to it, you can also do this. Vertebrates instead of vertebrata, circle mouths or jawed mouths. Jawed mouths are either bony fish or cartilaginous fish. Bony fish are either ray fins or lobe fins. And so a lobe-finned fish is a bony fish, is a gnathostome, is a vertebrate. But these things help us break it all up. And it's also an accurate reflection of the evolutionary relationships of all these groups. We've just made these nodes have clade names. All of these are monophyletic clades that are also very helpful and descriptive, which is very cool. Are we good? All right, we have to play around in here for a while now. And we're going to keep coming back to these clades because there are still cartilaginous fish, ray-finned fish, lobe-finned fish today, of course. So let's put that into our temporal context. This, right, is the modern diversity. And actually, I'll do this for a second. Oh, wait, it won't work because of the animation. Would have been fun. So you guys have already seen the time-calibrated phylogeny. Here's that vertebrata monophyletic group back here in the Cambrian. Conodonts, which are a stem cyclostome, go all the way back into the Cambrian. Here's all these jawless fishes. And I'm sure you can guess what's going to happen now. I'm going to take this previously collapsed gray bar, which was the gnathostomes, for the last 2 weeks or 2 lectures of class and show you the real horror of fishes. in the Paleozoic. And so here, those are all the gnathostomes that we can talk about existing in this early, middle, and late parts of the Paleozoic. You guys are seeing all the way from 539 million down here to 252 million way up there. This is the entirety of the Paleozoic. And so those same nodes totally map on the tree. There's Vertebrata, there's Cyclostomata. This is a little bit funny. We're about to talk about it. This is Gnathostomata, as some people mean it. I'll explain that in a second. Here's Gnathostomata, the crown group. So the crown gnathostomes are defined by the living groups, chondrichthyans and osteichthyans. There's a common ancestor they all share. But what's fun about paleontology is, well, actually, jaws show up in some of these fishes, but they're just all dead. So we have a gnathostome crown. Does that make sense? But then if we actually mean like you have a mouth with a jaw, it actually does go back here, which is cool. Not going to freak you out about that too much, but I want you to see it. And then within Gnathostomata, there's your osteichthyans, there's your chondrichthyans. Within your osteichthyans, there's your sarcopterygians and your actinopterygians, which is still collapsed. Don't worry, I'll blow it up for you later. It's a lot of stuff though, right? It's a whole lot of things. What I have on there now, sorry, are these pictures to help you make the connection with that phylogeny you've just seen of all the living actinops and sarcops and chondrichthyans. But in fact, it's more fun to do this. Here's the all fossils and organisms we know really well from these actual time periods of these different clades. So your magpie has now been replaced by an early amphibian-y kind of guy, because that's actually what's around in the Carboniferous, not a magpie. One thing that I think is really cool is there's all of these other jawed fish lineages that we're going to have to talk about. They've been recognized by paleontologists for a long time. They have a name. Both of these names reflect what we now understand to be paraphyletic clades. We can accurately describe their anatomy, but they don't have a common ancestor and then are their own thing. Some of them are more closely related to us than others, which is cool. There's two groups of that. One is called placoderms. These three groups are representing different clades of these fishes that get called quote unquote placoderms. And these three clades, one of which is very closely related to some, but not all, of the living vertebrates, are these things called acanthodians. So again, a lot of names. I know it's so many names. I'm only giving it to you so that you have some way to navigate, do your own Googling, and then as we talk about stuff. I've been blabbing, but I want to show you some of these fossils, because you might see that the gnathostome branch has always been this low on all my slides, all the way back here to the Silurian. But there are members of some of these lineages, maybe not a lungfish or a tetrapodomorph, but something, so it doesn't go yet in either one of those groups, like down here maybe, that we have all the way back in the Silurian. So in the second period after the Cambrian, we already have our gnathostomes. So I want to show you some of those early gnathostomes. Here are some of those fossils. I've been blabbing for a second. Entelognathus was really important. It was on the cover of a lot of journals when it came out just a few years ago. Look at that fossil with your neighbors and make some interpretations, because you have to kind of know what you're talking about, but I can show it to you. What do you see? You can kind of see some of the labels, I think. I like that it's got these like rings of like these bony plates all around its tiny little eyeballs. Nostril, eyeball, eyeball, way up front. It's not Secundambaspis, but it is kind of like giving that, isn't it, a little bit? And then the jaws are super delicate. These are the lower jaws right here. Here it is in ventral view, so you're seeing it from below. Here's the right dentary and right lower jaw. Here's the left part of the lower jaw coming together. So this, one of the earliest actually articulated good fossil animals we have that has its gnathostome jaw. That jaw is a very small structure on its kind of proportionately quite big head. And so this animal is called Entelognathus. It's in one of those clades that's called a placoderm. It's one of the earliest diverging ones. But what I think is cool, and this is a factor of like Earth history, is that these gnathostome clades, you can see there's a whole bunch of them. Several of them have lineages, have fossils, all showing up at a, not exactly the same time, but like geologically speaking, similar time in this late part of the Silurian. So about 420 million years ago, we're getting different things with jaws. Some of it is direct, Intellignathus. You can look at the jaws. Some of it's a little smushier, but I think it's fun. There's scales known from many, many places in the world that are referred to this genus, Augustolepis. What you guys are looking at here is like a little scale cut in cross section, so you can like see the internal structure. We're going to talk about these scales in a couple of slides. This is exactly the kind of scale structure that living chondrichthyans, sharks and other cartilaginous fishes have. And so there's loose scales from the late Silurian, but no like teeth or skulls or anything. Isn't that cool? So like, okay, somebody's around, even though I don't have a lot to show you. Some of the osteichthyans, we have really, really great fossils. This is another lower jaw, which like, by the way, how perfect is it to only have a lower jaw for an early Gnathostome? It's not like I'm like, yeah, it's a real like Sarcopterygian brain case, so we can infer it has a jaw. It's like, no, no, here's the jaw. You want to see it? Here's the jaw. And so this one has a great name, Megamastax. Sometimes paleontologists are good. Big chewer, big Megamastax. And so you can see there's like different tooth rows, different teeth set into different bones that make up this mandible. But this animal is really reasonably thought to be a Sarcopterygian. So not even just an osteichthyon, but a lobe-finned osteichthyon, a lobe-finned bony fish, 420 million years ago or so. And there's another one, Sparalepis. This is kind of what I was just joking about. Sparalepis, we don't have its face, but we do have its body, and we have what are the spines that go in front of its dorsal fins, pelvic fins, pectoral fins. And so this is the fossil right there. Also, very strong evidence based on animals that are known from later in time that Sparalepis is like the body of a different kind, probably, of Sarcopterygian, so a lobe-finned bony fish, all the way back here in this Silurian period. These are all sort of tied with each other for being the oldest gnathostomes. Yeah, Char.
So you mentioned that Megamastax was a lobe-finned fish, right?
Yeah.
And you said we only have the jaw.
Yeah.
So how do we know it's a lobed-finned fish if we don't have any of the fins?
Anybody feel like they could field that as a question besides me? Because I'm not making this explicit for you guys. What would be a reason I might be comfortable with other scientists saying they have a jaw that belongs to a lobe-finned fish? I don't have its fin. If some of the anatomical details of the jaw are exactly what we see in like later lobe-finned fishes, that's like the assignment these people might make. And then I could say like, okay, yeah, I agree. Does that make sense? Anybody else have anything to add to that? Because this is a great question. Please, I'm always going to blab, and then you have to interrupt me and be like, wait a minute, wait. Please do that. Yeah.
So we have more fossils of not necessarily Megamastax, but Sarcopterygians, but
So many.
later that are fully complete, and it looks pretty much the same.
Right now, I'm trying to show you guys the absolute earliest scraps we have. And as soon as we keep going up, especially into this next period, which we're going to talk about for a long time today, I got so many. We all have so many. And so we can go down as far as we can go. This is kind of one of the flaws of going through geologic time like chronologically, like I'm trying to do, is sometimes it's like, well, how do we know that? I'm like, well, you'll see. And that's good. I'm glad you asked. So what I think is interesting is that people doing field work around the world, going to new places and looking for fossils and new locations, are finding stuff like this. All of these animals have been named in the last 10-ish, maybe 12 now years. They're all from similar places. Most of them are from this place in China. So here's some really, really nice paleo art of like late Silurian China. Here's Megamastax. Here's Intellignathus. Here's, or maybe that's Sparalepis. That's Megamastax back there. And you guys can see all these like gross little conodonts swimming around in that water. And so we can kind of build out, right? Right now, the best evidence humans have of gnathostomes is like pretty diverse gnathostome clades from one spot in China in the late part of the Silurian period. The presence of all of these different ones, I would argue, pretty clearly implies that we should keep going down in the Silurian and look in more different places, because we'll probably find the earlier members of these different clades. So yes, I'm trying to show you right now the oldest ones we have. Let's talk about the Devonian though, because this is how I can make those inferences. So thank you actually, Trevor, for asking that question. So just like we did before, I can grab my Devonian PowerPoint box here and stretch it across. Here's Devonian time spread across gnathostome evolution. Talk to your neighbor. Well, actually, I should say vertebrate evolution. Talk to your neighbors. The Devonian is called the age of fishes. Agree or disagree? And be specific, please. Talk about that.
all sorts of different, you got all the early ones, really, really, you know, the ones that are super specific.
Yeah, what's there, like, so it's something that I would put up or I started it and then it actually back down. What is their say they're close to this house?
Yeah, I mean, that would. That would explain why. Yeah, well, the girls were just like stuff. Is there actually potential products? So I had more of this shit. Oh, so that much. And like also, yeah, those say that. Say, oh, you know, the rooms come up here.
Okay, so while you're talking about this age of fishes, I think you all, I hope I was eavesdropping, so good. This is time calibrated though. So you can look at these things, you can compare them to each other, right? The gray boxes are meaningful here. The gray boxes are as far back as we have good fossils. The black lines are estimates of divergence. And so you can take both of those pieces of information and talk to your neighbors and come up with some like observations you might have or questions that might come up for you as you look at these distributions. So go ahead and just make those gray and black line observations and what seems like something worth talking about. What things could you get after? So spend a minute on that, please.
Yeah, I don't big. Yellow. It's just there's turns out like what's name and see I'm sure this is one person and this is sad one. I see friends I don't know if it's selfish. It's like there's actually one being. I think it's enough stating that we're going to go back to somewhere. So you got the greatest these guys possible this is they're still that he is going what we know about people who got break in that's just curious. Yeah, I don't think it's like something completely hard. Oh, there it is. Yeah, that's less budget. Our service, our conference, yeah, with the rest of them all. So that's great. Still, it's like maybe like in live. They're making offense, so I guess that's important for visual integration sometimes. So are they the other kind of strategy? No, they are. They're all over there. Busy like kind of say like how big it's a plan. Why? It's interest. Okay. Thanks.
All right, so what are some observations you have or questions that might come from those observations? What are your groups talking about? Yeah, Dakota.
Like a lot of heavily armored. Goats think they're very good.
So a lot of these gray boxes stop at the same-ish place in time. That's something people were talking about. Something you might add to that, of course, because I'm not really showing you hundreds. You already have seen how there's like hundreds of species in some of these boxes. But you're saying like some of these are kind of heavy duty armor-wise. And so you're making an observation, well, it seems like all these armored stuff's going away at about the same time. Cool, worth interrogating probably. What else? Yeah.
Are the green arrows,
Yeah. No, so Megamastax and Sparleapus, the ones I showed you from the Silurian, they're indeterminate. I don't know if they're on the coelacanth side, the lungfish side, or the tetrapod side. So the node is back here. So maybe they're here, maybe they're here, maybe they're here. It's hard to tell them apart from the. We know they're Sarcopterygians, and then that's as good as we could get. Does that make sense? Okay. Same with these Chondrichthyans. I have those scales that are Chondrichthyan scales. I don't have them. Humans have those scales from the Silurian that are clearly Chondrichthyan scales, but I don't know whose scales they are from those later clades. Maybe those clades don't exist yet. I haven't diversified yet. What else? Yeah.
I thought it was interesting that we have some purely fossils for the ray-finned fish earlier than a lot of the other ones, and
The oldest Gnathostome fossils we have that are definably like part of, with high confidence, one of the living clades. There are pretty interesting actinopterygians from the Silurian. Which, I mean, maybe that's blow your brains because obviously a lot of this is fresh as of 20 minutes ago. But it's pretty cool. I've always personally enjoyed that the earliest tetrapodomorph fishes are older than the earliest like true elasmobranchs, which are what sharks are. Like there's fishes that are getting kind of this kind of thing before we actually have what I would call proper sharks. That's cool. Now, someone's gonna find fossils here someday, so we'll work it out. But yeah.
I was just gonna say, in counterpart to that, it's kind of interesting that the rest of the crown stones are discovered until Devonian.
Yeah, and so we do. It's a little bit complicated, right? Because we do have like some. We know we have Saccharophrygians from here. There's kind of indeterminate, right? Whenever I cartoonify everything for you guys and make these slides, I do those kind of things. But I appreciate what you're saying, and I agree, right? All into the Devonian, almost all these other clades. So this is cool. And anytime we see these diagrams, like please think about those kind of things. This is one of the major mass extinction events in Earth history. We're going to talk about it. Don't worry. And that's my other question for you. Because you're right here. Somewhere in this is where mammals are. I think that's cool. You don't think about mammals and iguanas together, but they are in that one gray arrow on this slide anyway. Okay, so here's those two characters you got on the, remember there's 8 Gnathostome characters, but here's how they're distributed on this new phylogeny. You've already seen this basically, right? The Osteostracans have pectoral fins and a dorsal fin and a heterocircle tail. And then right at the end of class last Thursday, you learned that Gnathostomes, the total group of Gnathostomes, have jaws and 3 semicircular canals and pelvic fins. So those all show up here. And then all these Gnathostomes have those features, but this node represents the bracket of the living jawed vertebrates, the living Gnathostomes. Okay, let's have cool, fun biodiversity time. So I'm going to show you all the placoderms, even though they are a paraphyletic grade all at once. There's several clades of placoderms. What I want you to know about placoderms is that they're some of the most like diverse and abundant vertebrates we have, especially from the Devonian. They have a lot of very different body plans. Very often, but not completely always, they are characterized by like pretty heavy dermal bone shields. So Gary briefly mentioned this to you guys on Monday, but there's different kinds of bones in our skeletons. The bones of your skull, they're called dermal bones because they come from that ectoderm, that outer tissue layer when you're an embryo, your triploblastic bilaterian embryo. The outer layer is the ectoderm, and most of your skull bones come from that dermatocranial layer, and most of the armor pieces of these fish's skulls are that kind of dermal bone. So we'll talk about that. And then there's other things that are really fun about them. So here's one of these early placoderms. It's on the slide here called Lunaspis, nice and flat. Some of the most absolutely abundant placoderms we have, some of the most abundant fossils on the world are animals in this clade right here, represented by this little guy called Thimelepis. Look at those little eyeballs. Super heavy armored. Teeny little window way up front where both eyeballs sit. Mouth is down here underneath. They kind of have these really cool, they're called, oh my gosh, arthrodires is like the, no, not arthrodires. I can't remember their other name. Sorry, I'm forgetting. But they have these kind of like really fun pectoral fins that are so heavily armored that it like makes like these little points. You can see it up here on this one. It looks like an insect kind of, but even the pectoral fin is armored all the way to the tip. And they walk around on the bottom, we think, on those pectoral fins, which is really cool. I just like this name. Another benthic animal living on the bottom, Brindabell aspis. The most famous placoderm by an absolute mile is often in like Discovery Channel shows or Nat Geo type shows, is this animal Dunkleosteus. Dunkleosteus is on the slide. This is one of the first, like people think of it as like first like really truly huge vertebrate apex predators. There's controversy about how large this fish actually was. Again, the bones we have of this specific species are restricted to those heavily armored head pieces. But these animals are incredible because, I mean, even the eyeball is armored. It definitely has gigantic jaws and interesting things happening up top. It does not have teeth. Those are actually like just the bones themselves that are kind of sharp and like shear past each other of its skull. So it's reconstructed as like having exposed bone up there in its face. All these gaps in the armor are where we can reconstruct really quite powerful jaw muscles. And depending on who you ask, at this tip and that tip, this might be one of the most powerful, like, newtons of power bite in all the vertebrate kingdom is like the very tip of a Dunkleosteus's jaws, because there's so much muscle coming down to this one teeny point, not like a mouthful of teeth, but just like a kind of a thing. One other thing that you guys might like about this animal, if this animal is indeed an apex predator of a whole bunch of fishes that look like this, fossils that they find in the same beds that they find this fossil are called regurgitites. It's like a spit out pellet of armor of like a bunch of fishes all together. So you can imagine this thing wolfing everybody down and then like coughing up an armor ball after it chomps everybody, which is pretty cool, right? So Dunkleosteus, back in the day, everyone used to think it was about a 30-foot animal because we have much smaller relatives of this, and people would just take them and then scale it up. A bunch of estimates came out a couple of years ago, maybe 2 years ago now, that's like maybe Dunkleosteus is just a giant like Pac-Man and it's only like a 16-foot head, but it's a lot of head. And a lot of people get mad about that on social media. And it's really fun because you can just be a scientist and enjoy it. You don't have to take a side. I think it looks better small, but that's my personal opinion. Something that I want you guys to always be thinking about whenever we have these biodiversity slides is how evolution's always going to like let vertebrates over time expand into different niche spaces. So some of these are clearly benthic, living on the bottom. They have different architectures of their faces. This is quite a big placoderm that's clearly, I would argue, a predator. And like I just said, we have some evidence that it truly was a predator. But a really close relative of this chompy placoderm is another one that doesn't have any sharpness to its face. And we actually usually reconstruct it as if it was like a big filter feeder, like one of the earliest like filter feeding vertebrates. Many, many animals today, you guys might know basking sharks or whale sharks. Obviously, there's things like whales. There's plenty of other like bony fishes though, like anchovies. Lots of fishes today take water into their mouth, pass that water over their gills. We're going to talk about that in a second. And they just have like right in front of where they actually do their breathing with their gills, they just have these things called rakers where they filter out the plankton as it passes over. That's like what a basking shark does today or a whale shark can do. These are these early placoderms, pretty easy name, Titan ichthys. It's a big fish. And it's really closely related to Dunkleosteus. So think about the ecological diversification of all of these clades. This one's a big chompy apex predator probably. This one's maybe doing more of a filter feeding through the shallows kind of thing. Otherwise, we would infer really similar body plans and all that, but totally different feeding ecology, which I think is really, really fun. Anybody have any comments or questions about some of these placoderms? My favorite part of this whole class is just showing you stuff like this. Yeah.
So, I think I remember reading a paper in like a scientific journal somewhere
Yeah.
about a theory about there being like some kind of algal bloom during the Devonian that like killed off some fishes.
Uh-huh.
Have you heard about that?
Oh yeah, we're going to talk about the extinction big time.
Okay.
Well, yeah, after you meet everybody, you have to meet everybody who dies before we talk about the death. You know what I mean? But you're right. We're going to talk about it. There are theories that are about that kind of thing. Anybody else? Yeah.
Do you think that Dunkleosteus has?
No.
No.
No, I don't. Not in any like I like this very much. I don't know if we call those lips or not. It's more just like the soft tissue. It's not like a lip like you, like a muscular lip. It's more just like some kind of covering. I mean, if you look at, you can see paintings of this animal going back almost 100 years, and like sometimes it's like raw bone, like Skeletor face. It's like that's probably not quite right. I like this reconstruction very much, where you can kind of see these things that kind of look like teeth, but it's really just the tips of the jaw, and then yeah, soft tissue you could see maybe going up. So like a layer right here. And there is evidence for there's tissue there. It's not exposed bone in the same way. But I do think it is exposed bone if you're in the mouth. There's not a tooth. It's like if you didn't have teeth, if your white dentary bone was like sticking out. That's what that is. Yeah.
I was kind of thinking about it. If they had teeth and they're trying to bite down on things that are like down with crazy armor, wouldn't it come out? So I mean, it makes sense as part of the job.
Yeah, maybe this is just an animal whose anatomy allows it. Remember that there's actinopterygians from just as old, and they definitely are going to have teeth. I haven't shown you that yet, but like there's plenty of things with teeth swimming around when these things are living their best lives. So maybe that's why these ones are the apex predators at the time, like what you're saying. I'm open to that. I would hear more evidence about that. Here's more placoderms because I just like to show you how many there are. Bothrialepis is a really, really, really important animal paleontologically. It has a really, really long record through the Devonian. That's a genus. You guys are Homo. This is Bothrialepis. There were at one point over 100 named species of Bothrialepis from 1, 2, 3, 4, 5, 6, 7 continents. We have fossils of Bothrialepis. So this was a very successful Earth organism. This is one of those ones that has those jointed pectoral fins that kind of look like insecty legs. But occasionally you get fossils that have some soft tissue, so you can see the head or circle tail. It's the lower ventral fin. You can see the dorsal fin strapped onto this like basically like Hot Wheel car armor. I always think of it as with like spider legs. But these animals are really cool. This last data point has one of my favorite placoderm pieces of biology. There are fossils from this one genus, Microbrachius, and there are fossils, and if you find hundreds of fossils in a slab and you do it a couple different times, you can get a good sample of the population, and like 50% of them on the posterior part of their armor have these like hooks, and 50% of them don't. And so those are inferred as like male and female of these placoderms. And so having some sort of internal fertilization, I'm going to show you guys chondrichthyan, like shark reproduction in a little bit. Sharks do something really similar to this. And so having genital dimorphism, which is, I mean, obviously you're going to have genital dimorphism, but having like a big spur like this that's really obvious, it's a bony structure, potentially to be part of the fertilization process, implies internal fertilization, which is really cool. Fishes today sometimes have internal fertilization, but oftentimes they broadcast their eggs and their sperm out into the water. So it's cool to get after some of that reproductive biology. Some of those really nice fossils of these animals are known from really particular places. This is a place that I've never been, but I'd really love to go sometime. This is way out in the middle of the Australian outback. It's called the Gogo Reef. All these rocks are the actual like limestone structures of that reef. This is like a late Devonian reef. And so paleontologists who work in Australia, they go out there in their summertime, which is probably, I guess, right now, I guess, to do the paleo fossil work and find fossils. I saw a talk about Gogo one time, and the way that they described working there is if you bring a bunch of people, you get one fish per person per day, perhaps. And so it's a pretty rich site because if you go, you'll probably find a fish. They just break open all these rocks and nodules all over the landscape and see about fishes inside. What's really cool about the limestone that these fossils come in is that you don't have to like use a tool to like break the rock. You can dissolve the limestone and the bones will stay. And so Gogo is really famous, and it's been on the cover of a lot of journals many times because the fossils, once you get them out, don't look old. They look beautiful. So this is McNamara aspis. This is a little relative of Dunkleosteus. And so you can dissolve away the rock and totally see all the little details of its skeleton, which is really cool for a fish that's 370 million years old. And you can get like really thin bones, really perfect. They're all in place. And so animals like this one are how we can make inferences about things like Dunkleosteus, which is way too big to ever get fossilized like that. This animal's from Australia. Dunkleosteus is actually, all of them are from Cleveland, Ohio. They're from near Lake Erie. That's where Dunkleosteus comes from. Really cool. One of the most important fossils from Gogo of these early placoderms is this one. They named it Mater piscis, the motherfish. Attenborough, David Attenborough. They did this on purpose. So this is a fish fossil, and they have soft tissue, and there's a yolk and an embryo that they infer as being like within this female Mater piscis developing. And so you can see how if you have a placoderm fossil where you think you have an embryo with a yolk and that's quite large and it's a single baby, interesting for a fish, right? One high input offspring, not just like 1,000. If you have a nice fossil like this and they're from somewhere else in the world, like in China, you've got the sample size to show male and female and internal fertilization. These things go together. And so I always want you guys to be remembering we have a lot of limits in the biology of different fossil animals, but people who work really hard and synthesize all the information we keep getting from around the world, often new stuff, we can start to get the biology in ways we might not expect. So things like internal fertilization, live birth, showing up in the Devonian, at least, I'm very comfortable saying. No mammals yet, of course. This is only fishes. Yeah.
We start seeing evidence of freshwater.
Definitely in the Devonian. I'm going to show you so many freshwater fish. I don't know about the Silurian. I'm going to say Devonian probably. Yeah, so some of these fishes are found in freshwater. We'll talk about them in a minute. Everything I'm showing you for the most part so far has been in the oceans. So that's actually a great question. All those early actinot or Sarcopterygians and Telegnathus, ocean, 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 ocean. Almost all this stuff is ocean, ocean, ocean. Yeah. Okay. What's cool, I think, about the placoderms is like we can, again, track those early members. So this is early Devonian, middle Devonian, late Devonian. So here's like Dunkleosteus. and how like that armor can be like specialized for like muscle attachments and to make those giant jaws really work. This is one of the last living giant placoderms of all time, and it's also one of the big ones, and it's also one of the famous ones. And so I just want you guys to keep in mind that a lot of these things we have these evolutionary scenarios for. On this figure, though, the thing that I want to make clear to you is that there's dermal skeletal bones. So this is dermal skeletons, like the pink are these type of bones. You have the same kind of dermal skull bones. We're going to talk about that. And then there's all kinds of what gets called on these fishes, which is very weird to think about for vertebrates, but it's true. They're endoskeleton as opposed to their like, we don't like to say exoskeleton, but like dermal skeleton, the bones on the outside versus the bones on the inside. And so these, the jaw cartilages, the fins, the girdles, the vertebrae, all of these guys up here in these fishes are made out of cartilage. And it's only the outer ones that are made out of what we would call bone. So we're going to talk about that because it's a really big deal, vertebrate hard tissues. So I have to bring you back to this. So Gary showed you this in lab on Monday. And I think this is really important for you guys because one of the things I really want all of you to take away from this course in general, it's a bone-based course, of course, because it's paleontology. And some of you have like medical aspirations for your careers, is that this unit, this biological organ system, the skeleton, has a lot of different developmental origins and is one very complicated integrated system. Bone is not bone. There's different kinds of bone. And so you learned this a little bit in lab, and we'll talk about it more, of course. But the three tissue layers that every deuterostome and every protostome has, with an endoderm, a mesoderm, and an ectoderm, all three of those original embryo tissue layers lead to different parts of your skeleton. And so the ectoderm leads to these bones on the outside of your skull. For you, it's only your skull. But for a lot of these fishes you've been meeting, it's all those external armor bones. And then you have a couple weird bones, like your kneecap and your clavicle, that also come from this layer. Your kneecap's kind of its own thing. Whereas the mesoderm produces almost the whole rest of your skeleton. The actual vertebrate skull, like brain case, your vertebrae, your ribs, your arms, your legs, your pelvis, your shoulder girdle, almost all your skeleton comes from this layer. But you also inherit, but these fishes definitely have, a gill skeletal structure that comes from the endoderm. And so the endoderm, remember, is the lining of tissue that goes on this, which is like the food passageway, the digestive tract. And so these skeletal units start as part of your digestive tract. These ones, most of your skeleton comes from the middle. Some of the bones you guys have on your skulls comes from this outer layer, the ectoderm. Again, keeping it a little bit medical just so we can tie it back in. I want to show you guys what that kind of means in practice. So up here is an image you saw with Gary. Most of the color coding here, I'm going to try my best to walk through with you, but it's consistent across these three vertebrates. A shark, a fish, like this bowfin. You've seen bowfins in lab now. And then like this is a chicken skull, but I gave you guys a sage grouse because this is Idaho and that's more fun. Pretty much the same kind of thing. And so here's what I mean. The purple, the brown, the blue, the red are consistent across these three. So you can see here that osteichthyans have a lot of these red bones. Those are those like dermatocranial bones, whereas the chondrichthyans, none of that. So here, what am I talking about? That mesoderm layer, that middle tissue layer, is where things like your neurocranium comes from. So your neurocranium is the part of your cranium that like truly ensconces your brain. You guys have a lot of these bones reduced or lost, but here's some of the names: odic, exoccipital, supraoccipital, ethmoid. You don't have to worry about these, but I'm just giving them to you. And on these two, it's labeled as orbit because it's just showing you where the eyes go. I made it a blue circle because I'm just trying to make it clear. That like shark skull you guys saw in lab, that entire like blobby structure is that neurocranium of a shark. And you can see it through the eyeball here on this bony fish. Other things like the endoderm, like I said, make that gill skeleton. You guys don't have much of a gill skeleton left yourself, at least that turns into bone. But all these gill arches, the actual cartilaginous rod that hold out the gill arches of a shark, go like this. And then I hope you guys did this in lab when you drew that shark. You guys all drew the shark gills, right? I haven't graded your or looked at your things yet. I hope you could see that like the shark's jaws are sitting there in its skull, and then right next to them, it's just like gill, 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 right after those jaws. And so gill arch 1 and gill arch 2 are homologous with what is now the jaws of something like a shark. And so you can imagine in like an embryo state, this is where the digestive tract goes through, and these gill arches are for breathing. Water comes in, water goes out. Maybe you evolve some kind of filter feeding stuff. That's cool. But what the gnathostomes do is turn those front two jaw cartilages gill cartilages into actual proper jaws, which is really cool. There are bones in a bony fish and even in a bird that are homologous to those cartilages in a shark. So a shark might have a palate quadrate. You guys all have a quadrate as part of your skull. A bird has a jaw joint between, like its quadrate and its articular. Those are two bones from that endoderm layer. You guys have those, but it's in your ear. We'll talk about it later. I know this is awful. This is the one that's going through time. I could start with mammals and go backwards, and you'd be like, I get it all. If I start like this, you have to keep going like, we'll get there. Don't worry. The other thing that I think is cool is that gill arch 2 in a shark has a bunch of names. There's a bunch of anatomy. We can call these things. A fish has also gill arch structure, but something like a chicken has pieces that are homologous with that, and so do you in your voice box, and in a couple other places. But these cartilages are homologous in a deep way with each other. Gill arch 2, gill arch 1, and these different vertebrates, which is cool. So this is gill arch 2, and that's gill arch 1. Awful in a chicken. It's easy in a shark. We'll get there. We'll connect them for you. And then those red bones are those dermal bones that come from that outer ectoderm, homologous with some of the same armor pieces we see in those armored fishes. The shark does not have that, and it's a big problem. We're going to talk about that here in a second. This is making sense. I'm really just trying to like weave something. I hope when I ask you guys a question about this, don't worry, it won't be horribly hard. You'll go back and like try your best to like cinch it all together. So I want to talk about these two different kinds of bone I keep bringing up, because bone forms in a lot of different ways, but here's like the two really big main categories. All the armor on these fishes that we've been seeing, and like this bone that you all have, was formed intramembranously. So what we can imagine happening is like in the ectoderm of a developing embryo, these little early cells, they're mesenchymal cells, you don't have to know that, go down and start putting out these collagens. And then you get these little blue cells called osteoblasts, so bone-making cells. And they just start pumping out the living tissue of bone. And then they spread and they spread. And as they spread, some cells get trapped in that matrix. Those cells get called osteocytes. Those are the cells that keep your bone tissue alive. 10 years later, you break your bone, it can heal itself because there's osteocytes trapped in there forever. But then when it breaks, they can help heal it up. And you kind of get this weird bone. It's called cancellous bone. Got lots of holes in it with like an outer layer of these cells to help the bone stay alive. This is like how we would have developmentally intramembranous bone. This is the kind of bone that all these armored fishes have. And like I said, these are the bones that are like your ectoderm bones. This one, only this one, which is weird. And like just your kneecap. Okay. Almost everything else in your skeleton is what is called endochondral bone. And endochondral bone only forms from mesoderm and endoderm tissues. And the thing that's really important, and you guys know this if you've ever dealt with a baby, is that the bone is preformed in cartilage. So these are these little cells called osteoblasts that make bone. And then as they make bone, bone cells get trapped. In endochondral bone, which is pretty highly evolved, you have chondrocytes instead of osteocytes. You have chondrocytes, which make a cartilage structure. And then within that cartilage structure, you start to get vasculature, and then these osteoblasts come in later. And there's this collar that develops. And so along, like in a long bone, like your femur, it starts in the middle and then spreads. And then you end up with like a long bone with two caps on it. You guys ever like eaten a chicken or something and like the ends come off really easily of the bones? Like when you're eating a chicken wing, there's like those caps that come off the edges. So like a chicken leg has three different pieces, the main shaft and then like the top and the bottom. And so again, this endochondral bone is much, much, much different than this. This is no big deal. Lay down a flat level of tissues, a flat level of cells, turn that into bone, you got a flat bone. If you want to make a really complicated bony shape, this preforming it in cartilage is super, super, super helpful. And this endochondral bone that only comes from mesoderm and endoderm tissues, that's one of these features that unites what I'm going to show you in a minute, these bony fishes. I know this is a lot of stuff. I like this. It helps me think of things. This is from like a mommy website, and it totally works. This is how your skeleton works in terms of being intramembranous and endochondral. The skull, jaw, and shoulder girdle are these intramembranous ectoderm bones. Everything else is from these endochondral bones. It's not my favorite image in the world because it's 4 femur, but you get it, right? So think about, you guys have ever had a baby or dealt with a baby? Babies have like soft parts in their skull. It's because those intramembranous bones are like slowly forming. And eventually they'll fuse up and meet each other. And that's kind of what we can imagine happening in these armored fishes too. Okay, I know this is a lot of like osteology kind of stuff, but I think it's important because we're seeing these vertebrate animals that are experimenting with all of this diversity that you and I can just kind of go out and look at in living organisms. So I want to just like tell you these different cartilage type, or sorry, different soft tissue or hard tissue types, because in the Devonian and in the Silurian, this is when vertebrates are kind of like consolidating their skeletons, and we have all this experimenting, making these really cool shapes. So cartilage is the most ancient of all these vertebrate tissues. Those like non-vertebrate chordates have cartilage skeletons. The lampreys today have very cartilagey skeletons. You guys know sharks and stingrays still have cartilagey skeletons. Later stuff makes bony skeletons. So here's a nice shark brain. Here's a kneecap. You guys have plenty of cartilage in your body right here, right here. It's tough. It holds its shape. It's not hard. Okay. Imagine the material properties of cartilage. And so here's some just like facts about it. It's a pretty like low metabolism. It can't really heal very well. If you like really like cut your nose or cut your ear really bad, it doesn't heal up super great. That cartilage can really easily damage. And it's mostly made out of this like collagen protein. It has a lot of water in it. There's no mineral crystals like bone might have. But again, this is like that oldest of vertebrate kind of structural tissues. And so it goes back way before vertebrates. Cartilage is an inherited thing we all have. Our endochondral bone, like I'm talking about, like the femur of this lady right here, this is bone in her, but it was cartilage when she was a young person. Okay. Dentine is another hard tissue. It's really similar to bone. It's mostly mineral, but there are a lot of proteins that live in it and a lot of cells that live in it. Oh, sorry, no cells that live in the matrix. For us, dentine, even though it's very quite similar to like the structure of bone, because it has no cells in it, it can't repair itself. So if you guys get a cavity, your tooth doesn't like heal because there's no living cells in dentine. But otherwise, dentine is really similar to bone. If you break your bone, it can heal itself. And so for us, dentine is restricted to our teeth. You only have dentine in this yellow structure. The white part of your tooth, it's white like bone, that is the dentine. What I think is important for you guys to know is that the exoskeletons of all these fishes have tons and tons and tons, if you really zoom under a microscope, of these little bubble-looking structures called odontodes that are made up of this dentine. Homologous, exactly the same chemically as like your tooth tissue. That's interesting. Hold on to that. There's that dentine in this like, you know, take a cross section from one of the little bumps on these fishes. This is like this dentine structure right there. Dentine is really cool because it can just grow at its base forever. And so we're going to see a lot of animals experimenting with dentine later in class. A lot of things that you guys call tusks are just totally uninterrupted dentine. Dentine is really soft. You guys know dentine by another name. Ivory is all dentine. So it's a tooth-based tissue in almost all tetrapods, but in early vertebrates, it's kind of spread all over the place. Also, you only get that from the ectoderm. Only that tissue layer can make dentine. All right, last one I want to give you is this one, which is enamel. You guys probably know enamel. It's on your teeth. It's the only place you have enamel. This is a really wild vertebrate tissue. It's amongst the hardest tissues any vertebrate can make. 96% of it is like mineral structure, like a crystalline lattice structure. There are almost no cells at all, if any, or no up here, cells in the matrix of enamel. And so you can imagine, again, your dentist tells you to floss and your dentist tells you to brush your teeth because if you have bacteria that are sitting next to your tooth for a month and a half and they dissolve that enamel, your body can't make more enamel. Once that enamel gets made, the cells die off when you're a baby and that's it. The enamel's gone if you lose it. And so that's why you have to get a cavity filled. This is basically like a crystal piece of tapping on top of your like semi, not really alive anymore, dentin either. This enamel's only up there in your teeth. What I think is cool is that, again, if you go back to some of these armored fishes, like here's Drepanascus here, and you look at the cross sections of these structures on its body, yes, there's dentine, and then there's also enamel, or this is an enameloid, it's quite similar to enamel, on the outside of the dentine. So I know this is a lot of like tissue talk and anatomy talk. I'm telling you that this fish is covered in what under microscope looks like teeth. like with dentin and enamel and all the details of teeth. That's what's cool to me. And that the dentine itself is actually quite similar to the bone that we have. That's what's important here and what I think is really fun. Mammals, we're going to see later in class. I'm just telling it to you now because I think it's cool to be able to call back to it. The interplay of dentine, which is soft, and enamel, like you can carve ivory, dentine is soft, and enamel, which is extremely hard, is what allows a lot of mammals to evolve really intense diets. And a lot of dinosaurs do it too. Here's a beaver. This is a beaver's molars. The high ridges you see are enamel, and the low troughs you see are the dentine. And so it's not all covered like your teeth are. The dentine and enamel are staggered. And then as that beaver's chewing wood or chewing grass all day, that enamel's tougher than the dentine, and it'll have like a really strong chewing surface. So we're going to see that later, but mammals really only play with enamel and dentine again. Restricted, restricted, restricted only to our teeth. So it's weird to think about animals that are all covered basically in those kind of structures. Again, only from the ectoderm. So the last thing I'll say is what I've been saying this whole time. You could take some animals today, here's a shark, and look at its skin under a microscope. I'm going to show you guys shark skin under a microscope on Monday. This is what a shark's scale looks like in cross-section. Here's if you're looking at like a patch of shark skin from above. There's a pulp cavity, a dentine structure, and an enamel covering on every shark scale. Have you guys heard that like sharks are tough or like they'll scratch you? Yes. You can wear shark skin if you're a swimmer and the water flows over you like these teeny, like not microscopic, but very tiny ways of interrupting the flow of the water so that you can move quickly through the water. Sharks are covered. That's why if you touch a shark ever, go to a touch tank, they're really rough if you go backwards. They're still kind of weird feeling if you go forwards and like petting them. They're covered in these scales. They're called placoid scales. And again, the thing I think is really fun is that anatomists knew this 100 some years ago. The skin of these animals is hundreds and hundreds and well, thousands and thousands and thousands of teeny little teeth. And so for a long time in vertebrate evolution, people wondered, all those jawless fishes with all their armor, cartilaginous on the inside, and then little tiny scales that kind of look like teeth, how do we eventually get these set into our mouths? And so here's a shark's body, a shark's lower jaw in cross section. So like there's a scale out there, but then here's what the teeth look like in a shark anyway. So it seemed obvious to a lot of people that the evolution of teeth and jaws is this really complicated interplay. You can imagine there has to be a lot of experimenting happening of eventually getting like the outside of your body attached to one of the cartilagey gill pieces on the inside of your body. I think that's really fun and really cool. And again, this is the thing I want you to see. A pulp cavity with a dentin structure coated in enamel. I've got that in my mouth. You've got that in your mouth. Sharks have it all over their body. Then sharks also obviously have teeth like we do. This was a bit of a crash course here. I usually do this hard tissue stuff in labs so that we can talk about it and then look at stuff, but I had to move it to today's lecture. Kind of apologizing to you right now because I just talked so much. You're like, why is he telling me all this? And I hope eventually we'll contextualize it with the cool fossils and it'll make sense. So thank you for being patient. Any of you have questions about this stuff? I had to shoehorn it, so I am sorry. Okay. Now let's talk about the jaw part. We just talked about the hard tissues. We just talked about the teeth for a second. Now let's talk about the jaws. So here's one of these skeletons with a neurocranium from the mesoderm. And here's a whole big gill skeleton from the endoderm. Cartilage, 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 cartilage in the shark. But we can see fossils like placoderms and living cartilaginous fish and living bony fish that all have these jaws. So how do we get jaws? I think we have a pretty excellent scheme now that's pretty well corroborated by developmental biology and by some fossils to help us understand how you go from something like a lamprey, which has that neurocranium and has that gill arch skeleton, water coming into that little lamprey, remember, coming out the gills. Maybe it's breathing, maybe it's doing some filter feeding, and how you turn this into jaws. And so from fossils, we can see, and from developmental biology, we can see that gill arch 1 and 2 become the cartilages of the first jaw, and then like that main jaw, and then there's like a second gill arch that's pretty involved. And then by the time we get to a shark, this is what I'm talking about with like the first gill arch being the jaws, where the teeth eventually settle onto it, and the second gill arch being kind of like structural. That's this green and purple stuff right here. And so, I don't know. I think I maybe over-explain myself when I talk about this, but I just always kind of like am pleased to imagine like little lampreys sitting on the bottom. Like I'm going like this because I'm imagining it's like breathing, and those gills are right here. And then eventually, you saw in Gnathos, that little early placoderm, the earliest jaws we actually have that are articulated. They're these teeny little structures on that Gnathos. They're very thin. The skull's huge, and there's these teeny little structures. And so imagine the adaptive moments when some of these little guys are starting to be like, with the gill, just a little, just enough. And what would be the evolutionary advantage of that? How would it quickly or not quickly maybe work out to be like, oh, having jaws is way better at catching food, processing food than not. But starting from a gill arch skeleton, those anterior parts of your gills becoming jaws. And then once evolution has those jaws, how natural selection could so quickly be like, oh, I got scales right here. Boom, boom. I'm slicing stuff now. And so you guys can imagine that seems so weird. And there's so many weird fossils. And that's what I think is so interesting and so fun, is imagine playing with all this stuff. I don't know why I have those. Oh, yeah. Sharks are some of the earliest diverging gnathostomes that we have today that are still alive. And one thing that's really important to remember about sharks is all this is cartilage and it's all unconnected. I mean, it's connected, I'm sorry, but it's unfused. You can't like swing your jaws around, right? Whereas a shark has a brain case, and then these jaws are loose relative to the brain case. They attach here and they attach to each other, but they have movement. And so imagine the adaptive landscape that is possible when all you have is like a gill skeleton under a brain case. So there's just like footage of one of our living chondrichthyans, a goblin shark, and you can watch it just like move its gills / jaws. They're really jaws at this point. They're not doing any breathing. But how great is that? And so imagining in these silly little old fishes how this might have worked out is like perennially a thing. When somebody gets a new excellent fossil of a gnathostome, it's like cover of a big journal because they're going to have another teeny little piece of information to help us understand this origination. Once gnathostomes evolve, you guys saw the slide, rapid adaptive radiation, tons of different kinds of jawed vertebrates really quickly, almost all at the same time. So when this happened in evolutionary history, in the paleontological record, we should be able to track this explosive diversification. Placoderms are part of it. So again, they're all spread out. What is this, Brandon? Forgetting. Oh, I see. This is that dermal skeleton. On the gnathostome side of things, not over here, not hagfish and lamprey, not their relatives, but on this gnathostome side of the tree, I had two characters here that like help with all of these gnathostomes, crown and stem gnathostomes. Talk to your neighbors for a second because I've been talking so much. Do you have any memory of what these two snake morphs are for like the gnathostome big group? Talk about that real quick. Anybody? I mean, there are two, so you might be like, I don't know which one he means, but people took notes, surely. Yeah, Carmen. Yeah, it's a dermal skeleton. So that's the armor of these guys. And then this one was lateral lines. They can sense the water, but don't worry about that one. I care about the dermal skeleton one. This is what I wanted you guys to see. All my blabbing brought me to this. So we were in the middle of talking about placoderms, these armored fishes. Over here in yellow, here's your little cephalaspis, your little osteostrachan, cutie pie, not a jawed vertebrate. Everything here has jaws. In purple is one of the osteichthyans. It's one of our relatives. It's a tetrapodomorph sarcopterygian osteichthyon. It is a relative of us. All the green stuff on the phylogeny are different kinds of those placoderms. Some of those placoderms are more closely related to us. Here's little Intellignathus than others. Intellignathus is also really old. So the fact that it's high up in the phylogeny, I hope you think is incredibly cool. All these other things are placoderms. In purple, I'm sorry the colors keep changing. I'm stealing from people's papers and they use whatever colors they use, but I'm trying to help you out here. In purple, Meckel's cartilage is that lower jaw gill 1 cartilage that like a shark has. And then, unlike a shark, but kind of like us, this is all the dermal bone with a kind of tooth, maybe, or no tooth at all, embedded in it. Here's a phylogeny of placoderms. Here's the anatomy and architecture of the jaw cartilage they have on the bottom, and then the bony thing that goes on top of that cartilage. Okay? I would call this crazy. Yeah, read both of those and then talk to your neighbors about it. And I want to hear what you think because I love this so much. That's why I've been monologuing at you. Okay, talk about it.
Gosh, I have thoughts. That's something that builds really wide view. I have like precipitation, so. Yeah, like earlier, it doesn't. As we get later, yeah. Then there's. So, something else. Yeah, that is different structures. So, like, did you go one of these all over the place?
It's two on the bottom left.
Yeah.
It's like a cheeseburger, the other one, it's like a portrait.
Yeah, I think about it. Also, the ones right above it. One of them.
That's how they eat it.
That's not the lifestyle. Also, just. So, yeah, the strength of the change, yeah.
What kind of observations are people making? Yeah,
I don't know if this is correct, but kind of as we move on.
we don't care if he's correct. Okay, go ahead.
It looks like the bone structure, the dermal bone, kind of starts surrounding or maybe protecting the cartilage. I don't know.
One of the things I like about this figure so much, I saw it and was like, whenever I teach for paleo, I'm putting this slide in, is because you have to imagine when this stuff is evolving, if you took a comparative anatomy class, it shows you what our jaw looks like, and then it tries to be like, so it must have been like this. It does ultimately bone all around that first gill arch structure. That is what we end up with. But what I like about fossils is that, like, you want to see a million things that didn't end up lasting, but definitely happened. And that's like what I like so much. Is this what you should expect from like messy, gnarly, real world fishes living their best lives in an evolutionary time frame? You're gonna have different kinds. This is like the cartilage wraps around the bone. This animal is not the ancestor of all the vertebrates on land today. This one is. And so what I like about this for you guys is not any one story. I like the fact that even within this one paraphyletic group called placoderms, you can see so much evolutionary experimentation for real. And so it's like, why would we bother talking about these things? Specialists need to talk about those things because they care about the interrelationships of these fishes. For you guys, I just want you to be like, wow, jaws to no jaws to jaws is a crazy step. And of course, it's not one step. And of course, if we get enough good fossils, we can be like, and it was such a gnarly mess. This ends up being what you do. But a lot of other fishes did a lot of different things too. That's what I want you to take away. Does that make sense? Anybody else want to say anything cool? Or what were you talking about? Yeah, Trevor.
Like I just think the variety of diversification that we're seeing here is like really, really.
It is cool. I'd snorkel. Would you guys snorkel? I like scuba and snorkel stuff. Here's the other thing that's really fun. You've seen the phylogeny a bunch of times already. That McNamara aspis, that early go-go, here's that go-go fish, the one that was so pretty when I dissolved the limestone away, when they dissolved the limestone away. It has these dermal bones, part of that armor. Not for all placoderms, but these placoderms, which are called maxillate placoderms, the green, the pink, the yellow, the other green, these are homologous with the actual bones that most of us do still have in our skull. So you have a maxilla and a dentary and a jugal. Well, guess what? So does McNamara aspis. It has a lot of other crazy bones that you definitely don't have. But some of these bones we can trace through evolutionary time. And so within the placoderms is where like not just like the presence of a dermal cranial skeleton exists, but like some of the details of it we still have, or in these early gnathostomes. The other thing that's so fantastic is that that's where sharks and stingrays go. We call everything over here osteichthyans. Here's the gnathostome crown. We call everything over here chondrichthyans. And so what does that mean? Basically developmental biologist, current like biologists working on, they don't care about fossils. They're working on the development of like the structures of these animals. Anatomists, paleontologists, everybody kind of is getting to the idea that like sharks, the lineage that leads to today's sharks and today's stingrays must have like lost all their dermal bones. What happened there? You guys know that in the Silurian, we've got little scales of chondrichthyans, but we don't have their faces. And so the only thing we can suspect is if Magnamar aspis and all these other ones have a maxilla, then sharks are like, I don't have any bone at all. It's all gill cartilage. Did they lose it? Because they're putting teeth on their gill cartilage. You're putting teeth on your bone. If you have a symposium at a conference, this is what they would love to talk about. It's really cool. And I want you guys to think about that. Bony faces, but not you. I like that a lot. Okay, we only have a little bit of time left. Let me give you, here's all of our gnathostomes, right? Within placoderms. Here's a little synapomorphy. It's some, but not all placoderms. And this synapomorphy is really important. It's those teeth. So some of the placoderms have teeth, some of them don't. And so it's within that placoderm group that you get this like outer scaly structure starting to anchor to whatever you're using as your jaw. It might be the cartilage. When you guys were babies, you had a little cartilage like this, and now it's up here. Or it might be the bone. If you're an osteichthyan or a chondrichthyan, it'll be a different thing that the tooth actually attaches to. That's cool. But the presence of teeth is like a mid-placoderm, for the purposes of this class, mid-placoderm synapomorphy. Enamel and dentin, enamel and dentin. Oh, go ahead. Horribly, Nucleostes is actually in this group, but we think even within this group, they lose the teeth. Sorry, but that is true, which is really fun. Anyway. I don't know if we have quite enough time to do this, but I'm going to show them to you anyway because we have to keep pushing forward. So these are these gnathostome relatives. They're really close to the crown group Gnathostomata. One of these groups might be in crown group Gnathostomata. They're these little guys called acanthodians. It means the spiny fishes. They're these little spiny sharks. They're not really sharks. They last for a really long time. They have this little tiny dermal skeleton that has like the kind of toothy structure. They have true jaws. And then the thing that I think is really maybe important to tell you about them, we only have like a minute. Some of these are some of the first freshwater gnathostomes we get, little schooling guys. Yeah, I'll stop there for now. Poor acanthodians, that's all they're going to get. So if you have any acanthodian questions, you can ask me later, but we have to do it. Okay, we'll do chondrichthyans and the rest of the crown on Tuesday. And I'll see you guys in lab to talk about arms and legs on Monday. Okay, do your take homes.